Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 155th seminar, and I call it a Burroughs Wellcome Fund Day. I successfully finished my 4 weeks of international trips to Spain, Portugal, and New Zealand to give invited talks, meet people for collaboration, discussion, and attend conferences. It was fantastic to meet many colleagues and friends while also enjoying some personal time for fun. Life should be balanced between work and break or life. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Tammy Collins. Tammy is a program officer at Burroughs Wellcome Fund, handling the career awards at the scientific interface, which are very relevant to this audience, especially young researchers. These career awards are very competitive and also aim to bridge advanced postdoctoral training and the first 3 years of faculty service. Obviously, the awardees are also very attractive in the faculty job market, so I strongly recommend young researchers should apply. Tammy, thanks so much for taking this important role for young generations and future leaders, and the virtual podium is all yours now, and thank you again.
Thank you so much. I'm going to share my slides. Hopefully everyone can see this okay.
Yes.
All right, great. I'm so glad to be here today. Thank you for the opportunity. As he indicated, I'm Tammy Collins, a program officer at Burroughs Wellcome. I've been here for about 2.5 years. And prior to that, I was the training director at the National Institute of Environmental Health Sciences, overseeing our overarching postdoc, postbac, and summer intern training program. So today, before I get into the details of the career awards at the Scientific Interface program, I wanted to give you just a little bit of background about Burroughs Wellcome Fund as a whole, because for those of you who may not be familiar with it. So once upon a time, there was a pharmaceutical company called Burroughs Wellcome, founded many years ago by Silas Burroughs and Henry Wellcome. This really grew in the 1900s and became what many of you may now know today as GlaxoSmithKline. An interesting tidbit is that this company was the first to actually create giving drugs in a tablet-based form. Um, the founders were very socially conscious, and in the 50s, they actually created a nonprofit arm of the actual pharmaceutical company called Burroughs Wellcome Fund. And then in the early 90s, a gift from the actual Wellcome Trust, which is located in the UK, allowed the Burroughs Wellcome Fund to break off and become a completely independent, nonprofit private foundation. So our endowment today hovers just below $1 billion, and we award between 40 to 45 million per year in grants, specifically to institutions in the U.S. and Canada. Um, the grants that we give out come from the return on investment that we get from our endowment, which allows us to be independent in the sense that, we're not reliant on government funding or donor funding and that type of thing. Our overarching mission is to serve and strengthen society by nurturing a diverse group of leaders in the biomedical sciences, specifically to improve human health by supporting education initiatives as well as powering research discovery in some of the research areas of greatest need of funding. Um, what you'll see with many philanthropies is that we're seeking to fill in the gaps and fund things that are very important but may be underserved by larger funding bodies in the United States, for example, such as the National Institute of Health or the National Science Foundation. To meet our mission, we have a variety of different program areas as well as competitive awards. And I won't be able to tell you about all of those today, but what I did want to show you that we do have more than the one I'll be focusing on, which is the career awards at the scientific interface. And I think this is the one that is most relevant to the synthetic biology community. So, in terms of the award itself, it's a postdoc to faculty bridging award, similar to a K99 R00. It actually predated that, and it's been around over 20 years. And this supports 5 years of training, the first 1 to 2 years of mentored training in a postdoc, and then up to 3 years in the independent phase in a tenure track position. All of the funding that we give is direct cost. So this is what you would have directly to support your lab or your stipend. And that's one of the things that when I came on board at Burroughs Wellcome, I really wanted to increase the stipend allowance that we were providing for postdocs. And so now we provide up to $80,000 in the initial year of CASI or up to $85,000 in the second year. And additionally, we also now provide up to $5,000 per year towards caregiving expenses. Individuals can have no more than 5 years of postdoc experience at the time of application, and international fellows are eligible for this award. And really, the hallmark of the award is that it's specific to researchers who are bringing skills from non life science areas such as chemistry, physics, mathematics, engineering, with the goal of focusing on answering biological questions at the scientific interface. So, unlike the K99, you can only apply once per year for CASI, but there are multiple phases. So, there's a letter of intent phase, in which individuals submit a 4,000 character essay, as well as a reference letter from your mentor. A subset of those are invited to submit a full application for phase two. And then a further subset of those are actually invited to interview. And if you want to learn more about the upcoming schedule, so the letters of intent will be due in September. Our RFP should be available sometime in mid-June, early July. Around November is when you find out if you are invited to submit a full application. Those will be due in early January of next year. And then we always interview in mid to late April, and then you get a notice of award in June. To learn more about the application and where you apply, I have a QR code here that you can reference. You can also see the current RFP because we're right now currently in the middle of our 2025 cycle and actually determining who we're going to invite to interview. Another thing I wanted to point out at this site is that you'll see a recorded video where I give detailed tips about applying for CASI along each stage of the application process, from LOI to the full application to the interview. And today I'm going to briefly touch on some tips for writing a letter of intent, because I think this is the area that I get the most questions on, that people are least familiar with. And what I did is look at a variety of letters of intent that were successful in making it to the next stage and looked at what were some of the commonalities of those. And what I saw in those were that individuals described the key biological question that needed to be answered. And then in the middle portion, spent a good deal of time providing additional background information to the reviewers to describe what needs to be accomplished to address that question, giving some of their aims, and then some degree of detail about the experiments that they would plan to do in order to address that question. And then they tended to end on the person describing what their future career goals were and emphasizing how their own interdisciplinary training positioned them to be the best person to address the research question. So that's really the key, one of the key aspects of CASI is the interdisciplinary nature of the award. and having interdisciplinary training. And then finally, really hammering home the overarching broad impact of the research in question. And that's one thing about CASI that we really are looking for is what could potentially be the broad impact. And we're also willing to allow people to take risks in their research. So in terms of a few additional general tips, we're really looking for how innovative the research is. Um, Oftentimes, what we might see is a new tool being developed, especially in synthetic biology. And so if that's the case, what is the specific purpose is being developed for, and what novel biological question is it seeking to address? Another thing that I wanted to point out with the career awards at the scientific interface is that we also have a special call out for applications that are addressing climate change and human health or artificial intelligence in MI. So your proposal doesn't have to be addressing these, but if it is, once everything is reviewed for scientific merit, if it is high up there in the scoring and it's addressing a priority topic, that might help tip it into the, moving on to the next stage. And as I said, we really are seeking to fund high impact approaches. At the same time, you want to be wary of being so ambitious as for it to be considered unfeasible. So what I wanted to do was just share with you some paraphrase comments from reviewers, and these are on the LOIs themselves in terms of whether they were a go and made it to the next stage or if they were no-goes. So for ones that made it to the next stage, you can see the general comments about, how innovative it is, how success would be remarkable, commenting about the interdisciplinary nature of individuals, how it's solving an important cutting edge problem or with cutting edge methods. And one of the things that I really saw with ones that were a no-go is the commonality is that they were vague. So here you can see the reviewers talked about, even though it was actually very well written and easy to follow, it was vague in terms of what they were really going to do. You see a similar theme here where they talk about, the ultimate goal is compelling, but there was little information about what experiments would be done, and the reference letter wasn't providing much. Another example, indicating how important the research is, but the proposal was vague and is claiming to solve everything about a certain area with computation and AI without really saying how. So that kind of leads me to provide some tips on which I think are applicable in any sort of grant writing scheme. So on one end of the spectrum, you've got things that are too vague, but that does not mean you should swing all the way over to the other side and have too much jargon such that a non-specialist audience can't understand it. And you really want to think about finding that balance in the middle where you're giving enough information for the scientists to understand why you're doing what you're doing, but not so much that they've got to go look up every word that you're talking about. And then finally, I wanted to end by just showcasing some examples of projects we've supported in synthetic biology. We've actually seen an increase in synthetic biology type applications. So these are just some of the titles, everything from altering metabolism to engineering organ development using synthetic genetic regulation, bioengineering mitochondria, creating sustainable food out of thin air, engineering models to control cell communication and self-organizing systems. And for some of the applications, in the case of, for example, engineering metabolism, the ultimate application was to have a sustainable energy storage. I can't go into the details of the mitochondria project, but suffice to say, the engineering approach to that could result in something that's pretty cool. Also, in some of the other examples I mentioned have to do with, making crops that might be more resilient to heat or drought conditions. Engineering to create foods from gases and microbes, and molecular programming using de novo designed proteins. So, my take home message that I want to leave you with is that we're a nonprofit foundation that provides grant funding through a wide variety of competitive mechanisms. And we're really seeking to support those areas that are important but are underserved by other foundations or larger government based funding bodies. Specifically, with the Cassie Award, we seek to support proposals with a high potential for broad impact, and we're also willing to allow applicants to take greater risks. I didn't go into as much detail on this, but I did want to indicate that nonprofit grants in and of themselves tend to be much more flexible than their government-based counterparts, and we have a streamlined, shorter application process. So that's another benefit for applying for nonprofit grants. And I'd like to just acknowledge my team, Eliza Garrett, Lemma Hadar, Russ Campbell, and Mono on our communications team, as well as Lou Muglia, our CEO. And then finally, I just wanted to leave you with some resources. Um, these are things that we've supported at Burroughs Wellcome Fund, a practitioner's playbook for building public trust in science and other domains, as well as a climate science legal defense fund. This is alluded to in this Tactics for Trust, which provides guidance to scientists around their work. And thank you, and really happy to be here today.
Thank you so much. This is absolutely very important and informative, talk. I believe, many young people will appreciate the information, and then I hope the number of, application increase. That is more job for you, but it's more fun with the more applications, I guess. So thank you so much. I also, we need to emphasize again the importance of the funding to advance science forward, because there are some uncertainty recent years or recent time. We should kind of move forward with the real funding, real investment in science. Otherwise, there's no bright future, I believe. Thank you so much. I really appreciate it.
Yeah, thank you for the invitation.
Sure. Okay, so now the main speaker of today with a longer introduction, Elena Musteata holds a double major in nanoscience and applied mathematics and is currently a PhD student in Jeff Tabor's lab at Rice University in the Department of Systems, Synthetic, and Physical Biology. For your information, Jeff Tabor is my office mate when I was in Chris Boyd lab and in UC San Francisco. Her work has focused on using synthetic biology approach to address diverse medical problems, including chronic gut inflammation, oral cancer, immune regulation, and MRSA infection. She has also collaborated extensively with Omid BYSS group, also at Rice University, to develop hydrogel encapsulation platforms for improved delivery of engineered bacteria to the gut. That means her work is really interdisciplinary. For her undergraduate research, she worked with Nathaniel Caddy at the College of Nanoscale Science and Technology in Albany, New York. Her primary project focused on engineering plasmonic-based biosensors to detect and diagnose Lyme disease. She also worked on developing virulence inhibitors to target the pathogen Pseudomonas aeruginosa. She has also worked at NIST, or National Institute of Standard and Technology, for 2 research internships. As a part of Dave Ross' group, she generated libraries for large-scale functional screening of protein variants to enable a data-driven approach for predicting protein genotype phenotype landscapes. Elena, thanks so much for your time today, and please take it away. It's all yours now.
Thank you so much. Thanks for the opportunity to be here too. Really glad to talk to everyone today. Let me share my screen. All right. Can everybody see my screen?
Yes.
Okay, awesome. All right, so I'll be talking today about engineering kynurenine biosensors to detect and treat gut inflammation. And as mentioned, I work in Jeff Tabor's lab at Rice University. And this information contains some unpublished work, so I'll just be presenting some kind of new exciting stuff that we're working on. So we're interested in this molecule kynurenine because it's recently been found to be important in a lot of immunoregulation in the human body. So kynurenine is a tryptophan-related small molecule, and as you can see, it plays a role in many different parts of immune regulation and many different human disease states. And our lab is especially interested in looking at inflammatory bowel disease, or IBD, which is characterized by these chronic, painful, inflammatory flares in the gut. These flares are really difficult to predict. People get them over time frequently and at varying frequencies, and it's therefore very difficult to treat in a timely manner. And on top of that, the condition is very challenging to diagnose, usually relies on very invasive procedures like getting a colonoscopy or a colon biopsy, which you could imagine isn't very feasible to be doing on a regular basis to maintain your treatment. So there's really this need to improve diagnosis and treatment of this condition, and leveraging new potential biomarkers like kynurenine could do that. So it was found recently that genes for different enzymes in the kynurenine pathway are upregulated in both forms of IBD. There's two forms of IBD, Crohn's disease and ulcerative colitis, and in both of them, these genes were upregulated. And furthermore, it's known that during immune activation, tryptophan metabolism through this kynurenine pathway is actually upregulated. The inflammatory signals stimulate the different immune cells to produce interferon gamma, which directly and very potently induces this enzyme IDO that drives this increase in tryptophan metabolism through this whole kynurenine pathway. And it's been found that when you treat IBD and when the symptoms start to be alleviated, these metabolites in this pathway actually return to normal ranges. So this kind of further supports the really important role of this pathway in this disease state and in inflammation in general. And most significantly, kynurenine levels in the serum increase during inflammation, and especially the kynurenine to tryptophan ratio increases. So it would be great if we had a device that could sense and respond to kynurenine levels directly in the gut so that we could get a quick snapshot of the patient's internal state and speed up that time to diagnosis, ultimately getting them faster and more timely treatment when it's needed. So we think synthetic biology could provide a great approach to this problem. Our lab and others have engineered probiotic bacteria that can easily be delivered to the gut to sense and respond to inflammation right at the site of the problem. So we want to develop a new theranostic bacteria that could detect this exciting new potential biomarker, kynurenine, as an input, and output either a fluorescent reporter protein for starters as a diagnosis tool, or an anti-inflammatory cytokine for treatment to lower the inflammation. And our design constraints come with unique challenges because we want to potentially down the line, this technology might want to be deployed in systems using animal models in vivo and also ex vivo for human fecal sample diagnosis. So when they get to the colon in an in vivo model, these designer probiotics would secrete IL-10 therapeutics only when they detect inflammation. And this would help inhibit the overproduction of interferon gamma and restore balance to this pathway, ultimately and hopefully helping to reduce inflammation. In the ex vivo human fecal samples, we would just obtain fecal samples And the bacteria could be deployed directly into the sample for what could become a rapid point of care test in the clinic to detect inflammation that would require very little technical skill and very little time to process that data. So it would be a lot faster than having to, get a colonoscopy or run a really long ELISA, which can take a while to get results from that back from the lab. So to do this, we first want to find a kynurenine responsive protein. So first we look towards nature for that, and luckily nature has provided us with lots of diverse bacteria and lots of different transcriptional regulators and proteins that respond to different ligands already with pretty high specificity and affinity. So one of them is not very well characterized, but has been described in some literature as kynar, and it responds to kynurenine. The specific kynar that we are using is from the soil bacteria, C. necator, which is a gram-negative bacteria, and it's really well known for its metabolic diversity. It has a lot of diverse ways to regulate gene expression. So this protein kynar was computationally mined from its genome, but it was non-functional when it was ported into other bacterial species, including in E. coli. So it looks like it was really only working in this soil bacteria. However, there's many problems with, keeping it in the soil bacteria. For one, there's a lot of cross-regulation that's occurring because in this bacteria, the protein controls a very large metabolic gene cluster, and its cognate promoter is also likely cross-regulated by a lot of different factors. And of course, it's not a suitable host for in vivo deployment to the gut. It's a soil bacteria, and it's not as well characterized as some other bacterial hosts that we would want to use. On the other hand, E. coli Nissl, everybody's favorite gut bacteria, is a probiotic bacteria. It's also gram-negative, and it's an ideal chassis for these gut-based therapeutics. It's actually been approved for human consumption under the brand name Mutaflor in some countries. And importantly for us as a design constraint, it does not metabolize or synthesize kynurenine, and it's genetically tractable. So this should eliminate kind of the cross-regulation of the kynar-regulated promoter, and should give us a better chassis for doing this kind of work. And just as a quick note on its safety and efficacy, there was actually a clinical trial where administering just wild-type Nissl was as effective as the standard treatment mesalazine, maintaining remission of ulcerative colitis. So this probiotic is already kind of a good chassis for this application where it already helps balance the gut. So how do we port this sensor from this species into this other species to make it more useful and tailored to our disease application? So first, let's look at the kynar gene in the genome of this soil bacteria, C. necator. In its native context, it's divergently transcribed from a large gene cluster, where I just show the first gene here. And while it's known to regulate expression of these genes, which encode for various enzymes that are needed to degrade tryptophan and kynurenine, its DNA binding sites within this region are unknown, and it's thought to also potentially auto-inhibit itself in its native context. Because as we'll see later, this protein belongs to a unique family of transcriptional regulators, the LRP class of proteins, that form these large octameric structures and bind long tracts of DNA. Which can make them auto-inhibit their own transcription by kind of blocking this whole sequence of DNA. Yes, as I mentioned, we don't really know where it binds right now. So to move this into E. coli, we started off with a few basic approaches. First, we had to decide what DNA sequence to actually use as the kynar-regulated promoter. And it's challenging to computationally predict operators for this class of proteins. Because they often lack the consensus palindrome sequences for typical regulators. And additionally, these factors are thought to recognize actually DNA structures more often than the DNA sequence. So altogether, this means we don't really have a good starting point to trim down this large region. So for starters, we are going to just use this entire intergenic region as the kynar regulated promoter. However, we did separate it from its promoter. We separated the protein expression from this native orientation so that we could eliminate any possible autoinhibition and improve expression in the non-native system. And we ultimately want to constitutively express kynar so that we eliminate any need to add any exogenous inducers for future in vivo applications, because when you're already starting with an in vivo system, you don't want to add extra complications and be delivering extra things in the animal model. And of course, we codon optimize this for expression in E. coli. So using this basic system, we first wanted to find what's the best expression level for the kynar protein. So since it's thought to function as an activator in the presence of kynurenine, we would expect that increasing kynar expression would increase the sensor response. So we constructed this inducible system where we can control the amount of kynar protein by adding the small molecule inducer ATC, which is here on the x-axis, and the fluorescent reporter is then activated by kynar. And as we would expect for an activator, higher kynar expression led to an improvement in the response to kynurenine. So next, we wanted to design a system more suitable for in vivo deployment, where it's not feasible to introduce these inducers like ATC. So we already knew that we would need fairly strong kynar expression. So we screened several already very strong promoters and found one here in pink that generated the strongest response to kynurenine, which is again here on the x-axis. And as a control, we also tested the system with no kynar protein at all, which is this black group. And we were able to confirm that the kynar regulated promoter is completely inactive in the E. coli system, which also supports it's not getting kind of cross-regulated or cross-activated by anything. So now that we have a working biosensor, of course, we want to break it. We want to generate an inactive mutant that can't bind kynurenine. This is for many reasons. First of all, it's helpful as a negative control in diagnostic studies, and will also help us better understand the protein structure, which isn't really well known. So as I mentioned, kynar is part of this interesting class of LRP type bacterial transcriptional regulators that form these octamers that can actually bend DNA and bind at 3 different sites. So the structural data on these proteins is pretty limited, and there's no structure for kynar. So one of our postdocs in our lab, Eric Wald, helped us out and generated a structural prediction of kynar here by alignment with a closely related LRP type protein. And from this model, we were able to predict the most likely ligand binding site and residues, and we cloned a few mutants to try to test some of these out. And luckily, actually, the second mutant we tested worked wonderfully and completely eliminated the kynurenine response, this W80A mutant, which is here in the ligand binding domain. And we think this inactive variant should really provide the best and most rigorous negative control for any future animal diagnostic studies or in vitro studies, because you would be testing or administering the exact same strain with just this one amino acid difference, rather than using a wild type bacteria or no bacteria as a control. So this should be very helpful to convince us that if we see any activation after passage through the gut, it is actually due to detecting kynurenine, and not to maybe many other confounding factors which can happen in a complex in vivo system. So we also wanted to make sure that this was pretty specific to kynurenine. So we tested the specificity of both the active kynar and the inactive mutant to other different kynurenine-related metabolites from that kynurenine pathway. And we found that indeed only kynurenine can activate the sensor, and the mutant sensor was also inactive across the board. So that's a good sign our mutation didn't increase promiscuity in the binding pocket or anything like that. And of note, tryptophan does not activate kynar, and none of these other metabolites activated it either. So since E. coli is not known to metabolize kynurenine, we were wondering, how is kynurenine entering the cell? So we hypothesized that it might be entering the cell since it's closely related to tryptophan, through some of the same transporters that transport tryptophan, which enters the cell actively using these 3 intermembrane transporters. So first is AROP, which is just a more general aromatic acid transporter that's able to carry tryptophan. There's also MTR, which is a high affinity transporter believed to be very specific for tryptophan and indole. And then TNAB is very specific to tryptophan, and this is the most critical transporter. It's responsible for pretty much all the tryptophan uptake. So we found that, first of all, it is likely that kynurenine enters the cell using these same 3 transporters. Here on this dark on the diamond group, you can see that this is just in the wild type strain. We see our normal dose response, and using these 2 knockouts that lack these 2 transporters, the response significantly decreases. And then when we have a knockout of this MTR transporter, we get completely no response at all, really strongly suggesting that pretty much all the kynurenine, or most of it, is being transported into the cell by this MTR protein, which was also really interesting to us because it's believed to be very specific for tryptophan and indole. So it seems like it might also be a kynurenine importer. So that was exciting to find out. And then during our continued sensor optimization, we also found that tryptophan actually inhibits kynar activation. So while kynurenine activation is typically very strong, it's highly repressed by adding tryptophan into the system. So you can see here on this heat map, there's kynurenine on the x-axis in increasing amounts and tryptophan on the y in increasing amounts. And you can see when there's no tryptophan in the system, the kynurenine activation is as we would expect. However, as you add increasing amounts of tryptophan, the activation becomes lower and lower until it's completely inhibited by 2.5 micromolars of tryptophan. And of note, tryptophan on its own has no effect on this system, and there's no basal expression from this promoter in the absence of kynurenine. So this is kind of cool because the effect of two ligand systems has been pretty well characterized in enzymes, but this kind of behavior is not typically reported in most transcription factors, likely just by virtue of the fact that these kind of activator experiments are not usually performed in the presence of these alternate potential ligands. So we also wanted to see, is this a specific property of this protein for tryptophan? And indeed, it seems like this inhibitory effect is specific to tryptophan. These other kynurenine-related metabolites do not inhibit the response. Only tryptophan takes it down from up here all the way down to like basal expression when you add it in at this 2.5 micromolar range. And this also makes sense if you think about the pathway that this protein is regulating, that actually the primary metabolite that it starts with is tryptophan. Kynurenine is the next intermediate metabolite. So it's really interesting that it might have been specifically evolved to respond to both of these proteins. Both of these ligands, sorry. So as further confirmation that this is indeed how the system is working, we wanted to check that the intracellular tryptophan levels are really what's directly affecting the sensor function. So we tested our system in different knockouts that are related to tryptophan metabolism in E. coli. So this plot over here on the y-axis shows the sensor activation at 200 micromolars of kynurenine, which is over here. So first we tested, just the base, which you can see the normal level of activation. And then we tested a TNAA knockout. So this protein is responsible for nearly all of the tryptophan degradation in the cell. So we would expect that in this knockout, the intracellular levels would be higher than normal since it's not able to degrade any of the tryptophan. And indeed, we see compared to the basal system, this knockout has much lower activation. Then these other 5 enzymes are responsible for tryptophan synthesis in the cell. So knocking them out should lower the intracellular tryptophan concentration and improve sensor activation because even that basal level of tryptophan in the cell has been lowered. And we do see that this activation is higher than expected. So this gave us pretty strong supporting evidence that this protein is specifically inhibited by tryptophan. So we wanted to see if this inhibition effect was a result of both ligands actually directly binding to kynar. So we purified the protein and ran isothermal titration calorimetry to check that both ligands are actually binding the protein. And you can see here that both kynurenine over here and tryptophan do directly bind the protein. And this is the first time this has been reported for kynar for either ligand, which is exciting. So we also wanted to look at kynar homologs from different species and see if this tryptophan inhibition property was conserved. So we identified kynar homologs from 3 primary species where they're most often found. So there's one, the kind of representative of like different genuses from where they're found. So there's the one we've been working with in Senecator, one in Burke-Wilderia, and one in Pseudomonas aeruginosa. And you can see that pretty much in all of them, the architecture in the genome is similar. You have the kynar regulatory protein divergently transcribed from the gene cluster that it regulates that has all these kynurenine-related degradation enzymes. And then in Pseudomonas, it also has this kinase gene that's a little bit further away in the genome, but it's also regulated, is also known to be regulated by kynar. And these areas in bold are areas that we used as kynar regulated promoter regions. So each sensor was ported into E. coli NISL in the similar context as before, and we were able to get them working in E. coli. You can see they work to different degrees. The Pseudomonas one using the promoter from the kind B region, so this region, actually outperformed all of them extremely well. So it seems like this definitely is maybe a much better and stronger sensor naturally. And we did find that each sensor is inhibited by tryptophan. So all of these did have a decrease in activation in the presence of tryptophan. So this just provides us even stronger support that, across evolution from all these different species, this kynar homolog has the same property kind of across the board. So for kind of future direction, something we're considering working on is working towards a kynar mutant that is not as sensitive to tryptophan. There's definitely benefits of having a sensor that's responding to different levels of the two metabolites, because as we mentioned in the pathway, they're both important and their relationship could be important to diagnosing. But in a biological system, you're always going to have a lot of tryptophan. So it's also possible that it might be kind of inactive across the board if it remains so sensitive to tryptophan. So we did some structural modeling and also a multiple sequence alignment to reveal these conserved residues based on comparing them to known ligand binding residues in LRP type regulators. And we identified around 10 residues that should be the most important. And we've been working on screening them to see if any of them reduce tryptophan sensitivity while maintaining kynurenine binding. And we found that some of them did actually reduce tryptophan sensitivity, but they also had higher basal activation for some of them. But for some preliminary results, we have the predicted binding pocket residues 135 and 136 might be more important to tryptophan binding than kynurenine, because you can see here mutating it to A, the two mutants still responded very well to kynurenine. From the white to the gray bar, there's still a lot of activation, so that's good. But then when we add tryptophan at 200 micromolars kynurenine, there's not as much of a drop as there is in the wild type system. So that suggests we might be able to move towards lowering or even eliminating some of this tryptophan inhibition to be able to get a sensor that responds only to kynurenine. So to eventually deploy, if down the line we or others eventually want to deploy this sensor in an animal model, we first need to think about delivery to the gut. How is it going to get there, and what is it going to do once it gets there? So in collaboration with Omid Vesi's group at Rice, we recently published a hydrogel encapsulation platform to enhance oral delivery. Because delivering free bacteria to the gut causes them to disperse widely, that you can imagine that would make retrieval and analysis pretty challenging, and their viability can also be affected because they have to go through the really acidic stomach and the harsh gut environment. So our new platform resolves several challenges associated with a conventional delivery of these free engineered bacteria to the gut. So we encapsulate our bacteria into this core shell structure where bacteria are loaded only into the capsule core. And as you can see here, our protocol preserves cell viability because we use gentle polymerization conditions and a biocompatible alginate formulation. And importantly, our system enables population-restricted cell growth within the capsules. So they always max that out around 10 to the 7 CFU, 10 to the 7 bacteria, regardless of the starting population. Our capsules also allow free size-restricted diffusion of molecules under 500 kilodaltons, which is suitable for detecting most biomarkers. And we tested one of our engineered sensors for thiosulfate, which is another proposed IBD biomarker, and found that our biosensor response was not affected by encapsulation. So this was just another sensor protein that responds to thiosulfate rather than kynurenine. And most importantly, this delivery method drastically improves sample recovery. So the bacteria are able to not only survive, but even grow within the gut. So you can see here before we gavage these, before we feed them to the mice, and then after we retrieve them from the stool, they've actually not only survived, but even grown within the capsules. So they give us this really high concentration and this really localized concentration of engineered bacteria that we can then analyze compared to just retrieving the fecal sample as a whole and having to deal with that really complex sample type and the low concentration of bacteria. So since we're able to get this cleaner and more concentrated sample, we can then perform flow cytometry and obtain single cell fluorescence values for only our cells of interest. And we do this by including a constitutive protein marker. So all the cells are going to be fluorescent red, regardless of whether or not they're activated. And then when they're activated, they also turn green. So we're able to not only retrieve this high density of cells, but we can also then sort out anything else. So if there's any kind of microparticles in there or other junk or other microbiota from the stool, we're able to pick out only our red bacteria. And we know that those are the ones that are getting activated. And importantly, we didn't have to discard any of the samples due to a low cell count. So these are some results from an animal model for DSS-induced inflammation, where we were able to see that in the healthy rats before we induce disease, there's very little green fluorescence, it's basal levels. And then after in the disease state, there's very high green fluorescence, suggesting that the thiosulfate concentration in the gut increased during the inflammation. And importantly, we also developed a method for disintegrating these capsules and retrieving the bacteria back from the hydrogels without killing them. So all of this together, we're able to administer these in an in vivo model without harming the bacteria, and we don't have to discard any of our samples due to low cell count. So it really makes the experiment more rigorous and easier, cleaner to interpret the results. So we're currently, again, in collaboration with Omid Vesi's group, considering deploying our sensor to diagnose colitis in a rat model, our new kynurenine sensor. So we would be doing a similar protocol as our previous study, where we induce colitis with a 10-day course of DSS, which is a well-established rodent colitis model. And we administer the capsules prior to and following disease onset. So we can use each rat as their own internal healthy control. And then after the gavage, we retrieve the capsules and analyze them with flow cytometry. So this is kind of a flow of the experiment where you just gavage the animals with the capsules and then retrieve the capsules from the stool and process them with flow cytometry after you're able to retrieve them. And just to kind of show some pictures, we were, as a first step, able to encapsulate our new sensor and get these very symmetrical capsules where the bacteria are loaded only into the core, and there's an empty shell layer around. And we found that the viability of our sensor was preserved for both the active and inactive sensor. The encapsulation did not affect viability, and it also did not affect sensor function. The inactive sensor remained inactive even in capsules compared to free cells. And the active sensor also performed similarly. So it looks like we can use potentially the same delivery method if we would want to, in the future, try to administer this to rats. So as a kind of last step, we might also want to secrete something therapeutic to help actually reduce the inflammation in response to kynurenine levels and provide like real-time treatment in situ. So there are several challenges associated with this. The primary one is that the cytokine must not only be produced by the E. coli, but you also need to secrete it extracellularly so it can actually reach its target. So after searching through many approaches, we decided to leverage this interesting protein, YEB-F, which turns out to be one of the only natively exported proteins from non-pathogenic E. coli. And the group that originally identified this protein showed that it was possible to use it as a carrier to export IL-2 outside of the cell as a fusion. So we thought that might be a good approach to try with our IL-10 cytokine. Another challenge, however, with producing bioactive IL-10 from bacteria when it's normally produced by mammalian cells is to make sure that it's still functioning and folding properly as expected. So a recent study from, I think, last year or 2 years ago now, computationally mutated a single chain version of IL-10 to improve its production in mycoplasma and to also improve its receptor affinity. So it's going to be more potent even if the bacteria are making less of it. And using this mutant, we were actually able to achieve for the first time kynurenine-dependent extracellular secretion of IL-10, and found that this mutant IL-10 was indeed secreted in far greater quantities than the wild type. So now, in addition to their protein having been tried in mycoplasma, we can confirm it also works in gram-negative bacteria E. coli. And we're also now running bioactivity studies on our secreted IL-10 to make sure that it's functioning as expected to activate the pathways. And potentially down the line, this could be tested in a mouse model to see if it does reduce inflammation in together with the inactive sensor, which won't be able to produce IL-10. So just as a general future directions, there's a lot of ways to leverage this new biomarker potentially to sense and respond to different disease states. So this is one biomarker. Calprotectin is another is the only clinically validated biomarker for IBD. So you can imagine a system where you would use kynurenine, potentially calprotectin, and potentially some other localization systems as well to make sure that this waits until it reaches the gut to actually activate. And you can use it, you can engineer different systems for logic and control to end up outputting your reporters or your dyes potentially, whatever. We have several ideas for ways that this could be more translatable for a point of care system as a diagnostic. And then for an in vivo system potentially to make some therapeutic molecules that would actually kind of close the loop and allow us to treat the inflammation so that rather than having to self-diagnose, I'm having an inflammatory flare and go to the doctor and get tested and get a colonoscopy. And by that time, like your internal state of your system might be completely different than it was when you first started. This will be able to respond like right away. You'd be able to see the change with these reporters and potentially treat it at the same time and without needing expensive purified proteins or expensive laboratory tests or clinical tests, which could make it a lot more accessible to a much wider range of people. And even for people to whom it is accessible, it's still very difficult to be able to do all those diagnostic measures regularly enough to track progression of your disease when you have these kind of chronic diseases with repeated flares. So this provides a way to kind of deal with chronic diseases in general to be able to keep track of them more regularly. So with that, I just want to thank my lab, my advisor, Jeff Tabor, and all of our lab members and our collaborators and our funding sources. Thank you.
Thank you. This is exciting work. Thank you so much. It's amazing. Also, I also realized how difficult making sensor is because we have also problem with the tryptophan interference because we start with the tryptophan sensor to develop some other sensor.
Oh, with like TRPR?
Yeah, actually we published one paper in Cell journal recently, but this is we develop multiple different sensor, but very specific to that compound, but tryptophan always kind of giving us some problem. Fantastic.
Yeah,
Go ahead.
Oh, I was just gonna say, yeah, tryptophan was really a little interferer. He's causing trouble.
Yeah, also a lot of, concentration is high in general, so that's another problem.
Yes, yes.
So I want to ask Tammy whether you have the comment or question before I go into the audience question.
Yeah, well, first of all, I was going to say I thought that was a great presentation, very clear, easy to follow. My question was actually it's more about Mutaflor itself, and in terms of the next stage of getting this into humans. Are you thinking of, I guess, ultimately those trials will be done in other countries, since the US is not allowing this, and also just was wondering what if there had been any discussion about the strategies around the FDA and getting them to be more amenable to this. And then the final comment I'll say, which I didn't really talk about, is that we actually have a program called Innovations in Regulatory Science to help overcome barriers in the regulatory process and help get things implemented for regulatory approval, as that's a separate program we have. So that was one of my main questions.
Yeah, no, that's definitely very important. And that's something we're thinking about. I will say for me specifically, my main scope focuses on really like the engineering, the sensor part of this. And I think there's definitely such good potential to really make an impact with this kind of technology down the line and to make these kind of monitoring systems more accessible to more people compared to the current standard of care. However, I do think it's definitely like a long ways off from getting out of the lab, from getting out of animal models. The part I discussed about with the human samples was like human fecal samples. So this would be done like in the lab, not going anywhere near a human. Something else we think about in our lab a lot is safety for down the line and however many years until these kind of technologies potentially become more well validated to the point where maybe they'll be applied and maybe they'll receive regulatory approval. But one thing we think about is, for example, biocontainment, even though they are approved in some countries already as being safe. You know, it's this new organism. So different ways we think about that is like a first kind of defense layer, like a physical containment. We actually have a different paper that hopefully will be coming out soon, leveraging our hydrogel encapsulation platform to provide a first layer of containment using a much tougher polymer material so that the bacteria are less able to escape outside. So that's already like a first layer of containment. And then a second layer of containment could be actually a genetic containment where you can make them dependent on certain things to grow or unable to actually proliferate in the gut. So that way you can be much more confident that even if the bacteria get out, they're not going to really like be able to grow or do anything. All that being said, they are already like, you know, we do believe that these probiotics are already pretty safe to use. And there's also different startups that are trying to administer probiotics and engineer probiotics. But from our point of view, we just like to think about, all aspects of that developing from the safety. And I think either way, even if they are already safe, improving the biocontainment can only help to get towards more of that regulatory approval and everything.
All right, thanks.
Tammy, do you have any other question or comment?
I think that's mostly it. Yeah.
Okay, so she also touched some of the question, but before I, ask that question, I also want to comment or answer some of the question Tammy already brought up. That's a very important one. Also, Elena also, answered beautifully, including biocontainment. My lab also working on the probiotic. We develop, multiple so-called kill switch for the biocontainment. But one thing I want to tell you is, interestingly, the strain she used, the E. coli Nisser, actually underwent regulatory process. Actually, unfortunately, however, they went all the way to the phase 2 clinical trial. Unfortunately, I don't know what is the reason, but some financial issue, some other issue, the company kind of experienced, and then they dropped the probiotic at the phase 3. It's kind of very disappointing one, but I believe the more company will be developing the probiotic and potentially some product out of the Jeff Tabor's lab, hopefully in the future. But that's a very promising one. And then they also consider biocontainment using oxotrophy. But US is somehow generous in terms of biocontainment. As long as you have some method, including oxotrophy, they typically okay with that one. So I believe, we will have some potential product out of this, synthetic biology, probiotic engineering, I believe, in the future. But thank you for the good information about the new program or another program you just, mentioned about regulatory thing. Okay, she already touched a little bit the question from Canyon, but I wanted to ask anyway one more time. How specifically are the cell population restricted within the capsules? Is it just a volume restriction? That is a question from Canyon.
Yes, we believe it's mainly a volume restriction, and also probably to do with the diffusion of media and nutrients into the cell. So that kind of restricts their growth. And we've tested it over like even 4 days, and it kind of remains constant. They remain viable, so they're not dying off, but they plateau at that 10 to the 7, which is kind of nice because then you know that the maximum amount of bacteria that you're administering.
Fantastic. one last challenging question from me. So having said the unfortunate, drop of that probiotic one, that also kind of indicate how challenging, making one scientific discovery into the translation. So I was thinking about that one for many, many, many years, but I want to hear from you. So what would be remaining challenges? You already touched some of that, remaining challenges for ultimate translation of such, technology going into the real, market. Can you kind of describe a few, and then how you're gonna overcome?
Yeah, sure. So I think, yeah, a big one is definitely safety considerations and regulatory approval. And I kind of mentioned kind of two approaches for that. Is yeah, first level of defense, having like a physical containment that really physically restricts the escape of the bacteria. Potentially also if instead of capsules, you would have some kind of like hydrogel implant, and then that's really easy to also retrieve it back out. So you can kind of pretty confidently say that you retrieved it all. And then in case it's damaged or broken in some way and the bacteria do escape, you have a second layer of containment with kill switches or oxytrophic bacteria, different ways you can control them so that they're unable to proliferate inside the gut. I think those two things should help with regulatory approval. I think there's also definitely like public perception is a big is important too. I think as with any new technology, there's always a balance between potential risks and potential like positive outcomes. So it just becomes a point to where for certain disease states, if it's worth the potential risk, and kind of seeing to what degree like the public or specific patient groups and specific like disease states it might be more worth for. From a design point of view, I think just ensuring very stable expression is important too. For example, genomically integrating these systems, making sure they're not dependent on antibiotics or other things like that, so that they're like stably expressed. It would be interesting potentially also to see like evolution over time, like over time on like longer scale experiments. Are our engineered systems stable? Are they genetically robust inside the cell? And I personally think another thing that could really like help move this forward is like minimizing the system as a whole. For example, for Nissl, we already know it's a pretty safe and good probiotic. But I think like potentially like removing more and more things to it to get to kind of like a minimal cell state where you're able to really say this is almost like just the biosensor. I think that would also further help convince people, it's not making anything else really that it would be making. It's really just doing, it's like we have more control over what it's doing. But it's already pretty safe too. So I think that could just be like further down the line, or if you're wanting to use other hosts potentially that aren't the same. Let me think. I'm sure there's lots of other
Sure.
restrictions too. I think also the delivery method is a big deal, which is why we're thinking about that. People don't really want to, other than all the technical design constraints, people don't really want to chug like a bacterial soup. Like it smells bad, it's gross. So definitely having a way to encapsulate it and deliver it more reasonably. And most of the studies with the engineering free bacteria, they do orally just gavage the like liquid bacterial culture, and the rats don't have much of a say in it, but it's potentially better to be able to have some improved delivery method to make it more palatable. And then, last thing from the output perspective for the diagnostic side of things, there's definitely, the fluorescent reporters are already great. We're able to like see this output, but my idea would be that down the line to really have like a point of care system, you would be able to produce some kind of dye or something. So it could even like stain, like stain a paper or some kind of like easy paper chromatography based test, or like stain the toilet water or something like that, so that you'd be able to really tell like, hey, I'm having a flare up, and then be able to like go take your treatment or take your drug or things like that. So just making it even more translatable with less technical skill required to actually process the results.
Absolutely beautiful. Thank you. Those things I always thinking about, but I confirm my kind of thought. I mean, I now wonder why I never collaborate with the Jeff, because he's my academic brother. So I just now wonder.
Yes, definitely.
And then you're my academic niece, right? Anyway, so thank you so much. I now see the 7 after 11. So let me close, and then we can chat a little bit more with some audience. Okay. So thank you all for joining and staying today. We'll meet again on March 13th, Thursday, the same time, the same Zoom link. We'll have Dr. Axel Bisser at Lawrence Berkeley National Lab, and also a deputy of science at Joint Genome Institute, and also Professor Christopher Dundas at the University of Georgia. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. Thank you. And I stop recording. Just give me 1 second.